this is meant to evoke Edvard Munch's famous painting, The Scream. And yet when I look at it, it doesn't look like a scream. It looks like sort of an expression of joy and delight. There's this inherent ambiguity and the emotion you might associate with this image. But much more deeply beyond this, what I love about this image are the beautiful pigments that it shows you that are pigments made by soil bacteria. So all of the colors here are coming from different types of bacteria that were isolated from the soil, and they're all making different metabolites. And you can see just visually that some of them stick very closely to the cells. They don't appear to diffuse very far, whereas others are much more diffusive. And so these metabolites come in a whole variety of colors. They have many other chemical properties. And yet I would argue that many of these metabolites historically have been overlooked with respect to what they do for the organisms that produce them. And the specific class that interests me are those that change color. Okay, and so if you look here in the upper part of this, I'm circling it, I hope you can see that with my cursor, a colony that has a darker shade of orange in the middle. And if only I had a time-lapse movie of this plate, what you would see is that that color comes and goes in time. So now let me just show you an example of this color change on the right with an organism, Pseudomonas aeruginosa, which is one of my favorite bacteria to study in our lab. And you see if you aerate this tube, the color changes now. You introduce oxygen, it becomes turquoise. You let it sit, and the color now switches back to yellow, which is the background color of the medium. And this can cycle again and again as long as you want. It's a fun thing to do in the laboratory. So what's happening is that in these cultures, there's a redox-active metabolite being made that's called piocyanin. You can see its structure here. So when the organisms produce this, it's in its reduced state, it's colorless. But when oxygen is introduced, it reacts with oxygen abiotically, transferring electrons to the oxygen and generating superoxide and being oxidized in the process, and then taken back up by the cells and re-reduced. And so this cycling is very important. And for many, many decades, this cycling reaction was recognized to be harmful, obviously because superoxide is being made. This is a broad spectrum, highly reactive molecule that can interact with all sorts of biological constituents from DNA to protein, etc. And this is why these molecules can be very toxic to the organisms that make them, as well as organisms in their vicinity. And yet that's not the full story. So back nearly a century ago now, a pioneering scientist by the name of Ernst Friedheim, whom I will point out was Swiss and came for 2 years to the Rockefeller Institute in New York to work with Leonard Michaelis in his laboratory prior to people figuring out even what the electron transport chain looked like. And this very thoughtful Swiss scientist recognized that these metabolites could serve what he called an accessory respiratory function. He called them accessory respiratory pigments. And he wasn't thinking about them in the context of bacterial biofilms. He was actually experimenting with what would happen when he added them to rat tumors. But what he saw was that it allowed the zone of the tumor to continue to do metabolism. He was calling it respiratory metabolism back then, even though he wasn't really sure what it was. And yet the point was that their electron transfer activity allowed them to do something physiologically beneficial. So this was a pioneering concept that sadly was forgotten and sort of totally ignored by the field several years after he published his work. And the reason why is because shortly thereafter, the golden age of antibiotic discovery essentially was kicked off. And this was through the extraordinary studies of Sir Alexander Fleming in the late 30s and 40s, and then Selman Waxman and others in the United States in the 50s, where these scientists were looking at the type of small metabolites made by soil bacteria when grown in proximity with others, and realizing that you could isolate compounds that had very potent properties against other organisms. And this, of course, led to the discovery of penicillin and streptomycin, and the rest is history. Now, what I want to emphasize is that despite the importance of this, clearly this is one of the most important discoveries in the 20th century for medicine. What was lost in the process was thinking about what these metabolites are doing for the organisms that make them. After Friedheim's initial work, it was really forgotten. And people began really focusing on the in vitro activity, the ability to synthesize these molecules and use them as agents of defense for medicine. But whether they were acting as antibiotics in nature was not something people were really thinking about so much until the late 80s, when another pioneering scientist that most people have never heard of, Dr. Linda Tomashow, did studies in Eastern Washington looking at the roles of particular phenosine carboxylic acid, in acting as an antibiotic in situ. And what she and her colleague David Weller demonstrated is that this molecule could inhibit the growth of a fungal pathogen of wheat. And so here you see the structure of this particular phenosine that's endemic to these fields that she demonstrated is actually made in nature at appreciable concentrations, and a lovely visual of the effect this has against the protection of the wheat from the fungus. So you can see here in the control on the left that the fungus in white is colonizing the wheat kernel. If this wheat is inoculated with a pseudomonad making phenosine carboxylic acid, it is able to protect the wheat against the fungus. And yet, if you only eliminate the ability to make this metabolite, then the fungus is able to take hold. So what she showed very clearly is that this particular metabolite, this particular antibiotic made in situ in these fields, is responsible in the biocontrol against take-all disease. And there's been a lot of gorgeous studies following this up afterwards. Now, we got interested in working with Linda a few years ago to understand whether or not the ability to make phenosines was distributed in lots of different types of soils. And we were curious regarding which types of organisms might be capable of making these molecules. And so a very talented former postdoc in my lab, Daniel Dar, who had great capability with bioinformatics, was able to develop a pipeline through which he could identify which organisms and which habitats were phenosine producers. And so what you see here is just a graph summarizing the bottom line of his search through, which was at the time over 800 metagenome publicly available databases, the distribution of estimated phenosine producers within these various habitats. And so in green are those that are colored as being from the rhizosphere, which is the zone of the soil in very close proximity to plant roots, versus in brown, more bulk soil. And while we can find phenosine producers in all of these habitats, they're concentrated within the rhizosphere samples. And that makes sense because the plant is giving carbon for these organisms, helping these organisms grow. And then in return, they're capable of making these phenosines. Okay, but this is just a very broad study. It's A, it's bioinformatic, and B, it's all done with bulk samples. And what we increasingly appreciate is that in order to understand what microbes are doing in any habitat, one needs to really begin to think about them in context. And the context is the microscale. And something that is very important, I believe, for environmental microbiologists of any stripe, whether you're studying the context of wheat roots or whether you're studying infections, is to think about how to actually visualize and quantify microbial activities in situ. And this requires technological innovation. So we began by applying methods that we had developed for clearing different types of samples, different types of tissue samples. In this case, we applied it to plant root. This is a wheat plant that we'd grown in the lab, and we're just looking, staining it. It's very beautiful. You can see these root hairs, and at the tips, colors indicating different little aggregate clumps of bacteria. I'm now going to actually flip into, for the rest of this talk, a context that is a different kind of environment. I would argue from the microbial perspective, it really doesn't matter which one you're in. Many of the soil bacteria that you find that are associated with plants are also ones that have the capacity to become opportunistic pathogens of humans. And out of convenience, sort of ironically, we have used the infected environment of individuals living with the disease cystic fibrosis as a habitat in which to begin to explore, how microbes are thriving at the spatial scales that we care about. And the techniques we've developed here, I think, are very broadly relevant. So I recognize Eawag isn't interested in infection per se, but what I hope you'll realize is that this is just one example application, and the methods and the questions are really very generalized. Okay, so if we were to zoom into this environment, what I'm now going to show you is a snapshot of mucus from an individual living with cystic fibrosis. In both of these habitats, I want to note that phenazines have been measured in bulk concentrations at appreciable amounts. Let's take a look at how the organisms are living. Okay, so we're zooming in, and here I'm going to stop it, and what you see is an aggregate of Pseudomonas aeruginosa living as a biofilm encased within this mucus. And we're about to now take away the mucus optically. And here you can see in blue human immune cells, neutrophils trying to come and kill this infecting pathogen. And then I'll eliminate these blue cells. And what you can see now is a different opportunistic pathogen in green. And the point is that these are polymicrobial infections. You have a variety of microbes in the mix. So what are they doing? How do they thrive? We need to know this if we want to treat these infections. We also want to know this just because it's fundamentally interesting. How is it that these microbes are able to maintain viability in environments like this? All right, so a prima facie question we can ask is, how quickly are they growing? This may seem like a very simple question, but it's actually a very difficult one to answer when you're trying to make the measurement in situ. And so this work was enabled by a super talented former graduate student of mine, Sebastian Kopf, who is a geochemist. And what Sebastian did, in collaboration with some colleagues at the Caltech GPS division, Alex Sessions and Victoria Orphan, was to adapt a method using fluorescent in situ hybridization, FISH, and nano secondary ion mass spectrometry to couple the species identification with a measurement of an anabolic activity. And so what you're looking at here on the left is a small snapshot of two aggregates in close proximity. And what we're trying to do is ask, how quickly are the cells in these aggregates growing? So the way we do this is to, prior to taking these images, incubate these materials with deuterated water. And then we calibrate how much deuterium is able to be taken up by these cells. And from this, we're able to back out their growth rate. And so here what you see is in the middle, a positive control that's just showing our ability to look at other ions that we know that they're going to have, certain ions of isotopes of carbon and nitrogen. On the right, we're now selectively imaging the incorporation of deuterium. And what you can see is that one of these populations is active and the other is not. So in the next slide here, what I'm doing is taking that image and we have processed it and quantified it so that you can have an estimate of the growth rate of those individual cells. And this is just showing a spectrum of growth rates we measured for cells from 4 different patients. And the important thing to note, there are 2 things to take away. One is that it's really heterogeneous, shockingly heterogeneous. The growth rates really span months to hours in some cases. And the other thing that I think is very important is that if we take the average growth rate is slow. It's less than 2 days. Now, this may not be at all surprising to environmental scientists, but this is something that, at least in the medical field, most people haven't really taken in, I think, as much as they need to, because a lot of modern drugs are still to this day designed against bacteria that are growing very quickly. And while this is a very smart thing to do if you're trying to combat bacteria that are infecting an individual, for instance, who has sepsis, their bacteria can grow quite fast and you want to knock them out when they're in that mode of growth. However, there are all of these important infections, chronic infections, where that is just simply not the case, where bacteria are growing much more slowly, and our current arsenal of antibiotics are really ill-equipped to deal with them in that state. So it's a very basic interest to understand what sustains microbes when they're growing slowly. So in this habitat, we can ask, what is constraining the growth? Why is it so slow? One can ask this in any habitat. And the answer in any habitat is usually complex. There can be many things that inhibit microbial growth. But one of the prima facie variables, I would argue, in any habitat, whether it's a soil or whether it's an infected lung, is oxygen. And I want to really dwell on this in this example because it's somewhat ironic. This is the airways. This is where we breathe. And so instinctively, we might be surprised that oxygen is limiting the growth of organisms here. And yet again, the thing is that we need to consider the micro scale, which is all that the microbe cares about. So first, let me just show you a calculation for steady state oxygen distribution that one might expect in a representative part of this complex pulmonary system. And that is of a bronchiolis with a diameter of around 500 microns. If we were to imagine it being filled in with mucus to different extents and take a slice across this tube, this is what we would visualize seeing. Okay, so from the top to the bottom, the mucus is filling it in. And then from the left to the right, we're going to, for this theoretical calculation, assume different concentrations of cells with the capacity to respire oxygen. And taking steady state calculations in mind, what we can come up with very readily is a distribution of what we would expect oxygen profiles to look like. And again, the answer here that is the important takeaway is that it's heterogeneous, that we would anticipate for this type of habitat that oxygen in some conditions, could still be replete, whereas in others you would expect large regions to be anoxic. And it is this heterogeneity that we need to reckon with as we think about how to understand the microbes and control them in this regime. And so this, you might recognize these type of plots are often shown in looking at microbes in sedimentary habitats, but you can do the same thing with microbes in sputum. So sputum is the mucus that these patients cough up. And just like you would drill down into a sediment, you can drill down into mucus with electrodes, and you can measure the microenvironment. And so this is just showing representative traces for sulfide, oxygen, redox, and nitrous oxide. And focusing on the oxic profile, again, the thing to emphasize is that there is a very steep oxycline. And so while within this, there's heterogeneous oxygen concentrations, a large fraction of the environment is quite anoxic. And this is true not only at the scale of millimeters, but this is really important at the scale of microns. And so for the rest of my talk, this is the scale we're going to be focusing on and really trying to understand what is sustaining microbes within aggregates when oxygen is depleted by virtue of the fact that the cells on the periphery consume it faster than it can diffuse. And this is my favorite slide in my talk. This was given to me by a former student, Scott Saunders. He was a fan of the Game of Thrones. This is a GIF from the Game of Thrones for anyone who liked that show. But I think it really depicts the existential struggle that these cells in the center are facing for nutrients. And it's a profound challenge. And it's a very interesting question to ask. How is it that they're able to keep going in the absence of oxygen, for example? All right. So they exist in these aggregates. Here you see again a snapshot from our images of the CF lung. And so much of our work is motivated by these spatial scales. And so in trying to figure out what these organisms are actually doing, we wanted to ask them directly. Daniel, this talented individual I mentioned earlier who did the metagenomic work, teamed up with his wife, who at the time was a technician in the laboratory of my colleague Long Kai, to invent a method that I think is going to be very powerful. And something I want to say is that Daniel is now leading his own lab at the Weizmann Institute of Science. He's in the Department of Plant and Microbial Biology there. And keep an eye on him, invite him for a seminar. He's just extraordinary. So what Daniel was able to do was make a new method that we call parallel sequential FISH that allows for the tracking of over 100 transcripts. That is the expression of genes at the single cell level in biofilm aggregates. So here you can just see the cells in black and white. On right, this is showing you a snapshot of how he did image processing, segmenting each of these cells so as to be able to trace the expression of genes within each of them and quantify them. So I'm just going to touch on this very briefly. This paper was recently published and I'm happy to answer questions later. But just to give you a snapshot of what it can reveal about microbial existence at the spatial scales relevant for microbes, I just want to show the following data. So again, let's start with a field of view. This is a view of Pseudomonas making biofilms very simply in the lab on a glass slide. Here you see them, we're staining for the 16S ribosomal RNA. Now, what you're looking at is those same cells that Daniel has classified based upon taking all of those 100 genes that are expressed in each and every one of these cells and classifying them using a statistical algorithm called the UMAP, which means the Uniform Manifold Approximation and Projection, which allows us to essentially convert transcriptional expression space into phenotypic space, where we're looking at patterns of expression that are distinct and then colorizing the cells accordingly. So that was a little bit overwhelming. And to make it more clear, now I'm just showing you 3 different classification groups in blue, yellow, and in red. To give you one example from the yellow group, this is a group of cells that are not only doing denitrification, but other things. Denitrification just is one of the many things they're doing. What you can see is that if you look at your favorite denitrification gene, for instance, the near S gene, in the raw reads on the right, it maps to where the UMAP cluster is in the center. And then to give another example, and to help me transition now to the rest of the theme of the talk, I'm going to return to these beautiful redox-active metabolites, phenazines. And these are 2 different genes that are involved in their biosynthesis. And what you can notice is that these genes are being turned on everywhere throughout this biofilm at this stage. So the point here is simply to take in that we now have the tools to interrogate microbes at spatial scales that are relevant to them, to learn, listen in on what they're actually doing. This is going to open up, I think, really important new insights into microbial ecology in many, many diverse contexts. And happy again to talk more about this method later. But to get to the scientific story that I think comes from doing this type of work, we can ask, why is it that phenazines are everywhere at this stage? How does this relate to what I have been telling you about in the first portion of this seminar, emphasizing the point of heterogeneity and the limitation for oxygen? Well, I think there's a real story there, and now let me tell it. So we know and have known for many decades now that as microbes grow in batch culture, when they hit this phase that's called stationary phase, they're running out of nutrients. And one of the nutrients that they're running out of is oxygen, because they're hitting a cell density again, where even though it's in planktonic phase, the cells are consuming the oxygen faster than it can enter the system. And what has been known now for quite some time through work of others is that in organisms that make phenazines, they time their production of phenazine synthesis to the point when they're just starting to run out of oxygen. And this is because it's regulated by quorum sensing, the ability to sense the population density. So here you see 2 of my favorite phenazines, piocyanin in blue and phenazine carboxylic acid in yellow. And a question that we've been asking in my lab now for quite a long while is, does this timing reflect something about their physiological function, hearkening back to Friedheim's early work that argued that maybe they could serve as accessory respiratory pigments? My first student and I, Mario Hernandez, took his pioneering insights and just asked them in the context of microbial biofilm, because we thought, well, that actually could make a lot of sense, that if these metabolites are somehow helping the cells when they're running out of oxygen, boy, it would make a lot of sense that they would produce them at this time. So many years went by, many people in my lab helped work on this idea. And sure enough, it turns out that phenazines indeed play a very important role in survival of these organisms as they hit this period in their existence where they run out of oxygen and other things, as I'm going to show you in just a moment. So just to summarize that and help now really sort of unpack those earlier images I was showing you of aggregates, where I was telling you on the periphery there's oxygen and then inside there isn't, let's just figure out at the cellular level what's happening and how the phenazines are relevant. So in blue on the left, this is zooming into the membrane structure of a cell that would be in the periphery of these films, one that has access to oxygen, that can grow really quickly, doubling once every 30 minutes. This is typical when you grow Pseudomonas in a laboratory. And this is just a depiction of its electron transport chain that looks very similar to that in the human mitochondrion. I'm not going to belabor the details. Suffice it to say that the passage of electrons through this chain, coupled to the translocations of protons outside of this membrane, enables an electrochemical gradient to be generated that powers the synthesis of ATP. This is classic oxidative phosphorylation. However, if you go into the interior of these films, where they're growing much more slowly, doubling on the order of once every 10 days or so. A very different physiology is going on. And multiple people in my lab have contributed to illuminating our understanding of this. We're actually now finally really getting into understanding it through the work of my current student, John Zemniecki. I'm not going to get into all the gory details of this. The point is simply that these metabolites of the phenazines can be cycled outside of the cell, where they react with a variety of things, depends on the context, oxygen in some cases, iron minerals in others, electrodes in others, allowing them to be regenerated and taken back up. And through this cycling, here's the key point. What it enables is a different mode of metabolism intracellularly, where the organisms, because the phenazines allow them to achieve redox balance, they are able to feed forward in a metabolic pathway that does what is called substrate level phosphorylation, where ATP is generated through reactions in the cytosol, and then it actually gets hydrolyzed at the membrane in the reverse direction from what the ATP synthase does. Here, it's hydrolyzing the ATP, allowing for the generation of this proton motive force across the membrane that can power other things the cell needs. So this is a more chilled out metabolism, if you will, but it works. It allows the cells to survive, doubling slowly, and it allows them to keep an energized membrane. And I suspect this mode of metabolism is quite widespread. I'll get into that in a little while. Okay, so this now gets me to the word agathokakological. So having built up to this, the idea that there are these gradients of oxygen in all these different contexts, from the soil to the clinic, that microbes sense this and they respond in kind, and yet their response is one that traffics in molecules which themselves have the capacity for both good and evil. How does this come together in our understanding of the development of these communities in different contexts? All right, so I'm going to very briefly just give you highlights of a couple of very recent papers, and then I'm going to go into depth in one story that really illustrates how the duality of the chemistry really plays out. Okay, so one brief highlight to share is work of two phenomenal students in my lab who've recently graduated. Lucas Morales, who's on his way to Switzerland actually to work in the lab of Alex Prusat at the EPFL, and Elena Perry. This dynamic duo decided to, recognize and pay homage to the fact that indeed these molecules can be toxic in the presence of oxygen. This is still very much a thing. And yet the new twist that they brought to it was asking if these molecules have been made in the soil for, millions of years by pseudomonads and other types of phenazine producers that we know are abundant in all sorts of habitats. Might there be a consequence to this, an evolutionary consequence to this that's relevant to our thinking about how microbes are able to be resilient against synthetic drugs that chemically look like them? And the answer is yes, that what they were able to show is that the defenses that these bacteria have evolved to handle their own natural antibiotics by making efflux pumps, by being able to respond to oxidative stress, gives them a really strong comparative advantage when they're hit with clinical antibiotics like ciprofloxacin. So I'm not going to take you through that data. I just want to highlight it because I think it's an example of taking an ecological perspective, thinking about the natural history of where these molecules are made and its consequences for how it is that these chronic pathogens are so resilient to the type of drugs we give them. In some ways, it's not at all surprising because they've evolved the ability to handle their own drugs. So when you see another compound that looks like the drug that you make, you're able to cope with it. This has important clinical implications. On a different note, and this one I think is an example that this community might care more about in some ways, I want to emphasize that it depends what is outside the cell, what these phenazines are able to react with. It's not only oxygen. We've shown prior to this recent paper many years ago that phenazines were able to reductively solubilize iron minerals. What a current postdoc in my lab, Darcy McRose, super talented. She's on her way to be an assistant professor in the Department of Civil and Environmental Engineering at MIT, showed is that she put two facts together, which is that the ability for phenazines to reductively interact with iron oxides can allow for the liberation of phosphate, which this community I'm sure appreciates can stick very well to iron oxides. What Darcy recognized was that the regulation of phenazine production is triggered by phosphate limitation. And this is also true for many other types of redox-active metabolites. You can actually see signatures of regulation by phosphate limitation in the genomes. And so what she showed very nicely in her recent work was that the production of these so-called antibiotics is regulated by phosphate limitation and can alleviate that limitation. And we think that, again, this might be a more generalizable phenomena. Happy to talk more about that later. But now for the rest of the seminar, I just want to tell you this final story where both the good and the bad come together in helping us understand a really important biological phenomenon that's broadly relevant. And that is the example of biofilm development. And here is a recent paper where we're talking about the implications of this sort of duality to the development of biofilms in the rhizosphere. Again, an environment that I suspect may be of interest to some watching this talk. So you can find all of these references on my lab's website to follow up. Okay, so here's the history of how we figured this out. So we've been interested in biofilms for a really long time. We've been interested in phenazines, and we started taking a closer look at these films to try to think about how it was that the phenazines were actually cycled within them. So at the top, what you see is data showing that these biofilms, this is now where we're growing them in the presence of flowing nutrients under a confocal microscope attached to a glass slide, are able to make, for the wild type, sort of mushroom-like structures. When we make it so that they're unable to produce phenazines, they can't form these big structures. But then when we add back exogenously one phenazine, thiocyanin, it restores that capacity to a large extent. If you zoom in, what you see is here on the bottom panel on the left in blue are the cells expressing a constitutive fluorescent protein. In the middle, you see a stain for extracellular DNA, eDNA, and then on the right, you see the overlay. The fact that there's a ton of eDNA in the matrix of these biofilms is not surprising. This has been documented by many, many laboratories, several of which I list here on the bottom. But what the function of this DNA is something that I think the field is working towards understanding. There are multiple ideas for what it does. One important one is it serves as a structural support. So when we think about biofilm development, we can think about it in different stages. And what I'm going to argue is that the reactions of phenazines at different stages as a function of how much oxygen is present in the environment allows the population to thrive in the long run, and that we need both the bad part and the good part in order for the entire population to survive. Okay, so stage one is forming what we call a microcolony, where we have individual cells attaching to a surface, aggregating and forming a microcolony, and then over time, that microcolony grows into a larger, thicker biofilm aggregate. Well, this first part is one that others prior to us showed is facilitated through the lysis of a fraction of the population, allowing that eDNA to be released and forming a scaffold upon which other cells can climb and begin to aggregate. What we suspected, based upon our earlier work showing that phenazines can sustain metabolism in the absence of oxygen, is that the eDNA might actually help in that process by retaining the phenazines and facilitating their electron transfer. So this was a very different idea for what the eDNA might be doing. And they're not mutually exclusive ideas, they're entirely complementary. So to pursue this, what we needed to do was figure out a way of selectively inhibiting biofilm development at different stages. So how to do this? Well, this returns to why I started my talk with showing you soil, because that's the natural ecological context where phenazines are made. And so in our effort to attempt to find something to selectively perturb phenazine production, we reasoned that we could leverage the ecological context and find an organism that eats these phenazines, figure out how it eats them, purify an enzyme that it uses to break it down, and then add that as our tool. And so that was what we did. So here you can see in the middle a plate of bacteria. This is classic environmental microbiology, which is super fun because you can select for exactly what you want by providing it as your sole carbon source for degradation. So here we are providing phenazine carboxylic acid. It's yellow. Here where the cells are growing, you can see they're depleting the yellow background. And I actually kicked this project off several winters ago and then handed the strain to my former postdoc, Kyle Costa, who's now on the faculty of the University of Minnesota. And Kyle was able to really figure out how it worked. And so what he did was, using a combination of genetics and biochemistry, identify the organism that made this, figure out the genes and the proteins. And to make a long story short, he focused on a particular enzyme called POD-A, which stands for the piocyanin oxidative demethylase, that catalyzes the demethylation of piocyanin, releasing formaldehyde and making reduced 1-hydroxyphenazine. And this is a very powerful tool now that we can add to biofilm cultures to deplete piocyanin from the mix. Here's what the enzyme looks like. It kind of reminds me of a ninja star. It is a trimer. And if you look at any individual barrel here, what you can see in the middle is the phenazine that's solvated in the catalytic core. And I'm not going to go into the details of biochemistry. All I'm going to say is that we know how this works, and so we can make mutants that are able to form their fold properly but are catalytically silent, and we can use that as a control in our experiments. So for instance, this glutamate residue here, we can mutate to an alanine and it can render this inactive. So using this tool, we can now do what I said we wanted to do, which is add this enzyme and ask, can we inhibit the early step of biofilm development? So what you see here is evidence that that's possible. On the left is what happens when we add the inactive version of POD-A. You see confluent growth. These are cells stained here in DAPI, so they're blue. If we add the active version, you see a large diminishment in the surface attachment. And we can quantify this here by surface coverage. And what we find is, I'll just walk you through it. The wild type is here on the left. The phenazine mutant is the next one over in white. And now here's our data with the enzyme. If we add the inactive enzyme, it looks like the wild type. If we add the active enzyme, getting rid of piocyanin, it looks like the mutant. And importantly, when we add either the inactive enzyme or the active enzyme POD-A with DNAs, it looks the same. And so what that is telling us is that this piocyanin and the ability to make the extracellular DNA are in the same pathway. And so you can either get rid of it by getting rid of the phenazine that's triggering lysis, or you can get rid of it by just adding the DNase and chewing it up after it's been made. In either case, this leads to an abrogation in biofilm development. So that was really exciting. I can come back later and take a detour and tell you the clinical applications of this, but for the sake of time, I'm just going to jump to how we're now using this knowledge that phenazines are really important in establishing these biofilms to explore what happens at the later stage of development. But there are very important clinical applications to treatment that I would be delighted to show you later. All right. So to get to the next stage of biofilm development, we can ask, what happens to the phenazines as these colonies are growing larger? And two very important questions that we were never able to really satisfyingly answer were, why is it these phenazines don't just diffuse out of these films, leading to their loss? And how is it that they can be recycled quickly enough to sustain the metabolic needs of the cells within them? And the problem was that we didn't have data quantifying how many phenazines were present and how quickly they were diffusing. So this was the goal of my former student, Scott Saunders. So the first question that he asked and then answered was simply, what are concentrations of different phenazines in these films and how are they retained within them? And then he asked an even harder question, which is, can he come up with a way to measure the rate of phenazine recycling within the biofilm? In other words, extracellular electron transfer. And is that rate faster than the loss of these phenazines from the films? Okay, so let's start with the slightly easier lift first. So what Scott did was use biofilms growing in a simple way to begin to address this question. Here, he's simply growing them on the surface of a nutrient agar where he's separating them with a permeable membrane. And so he can quantify the biofilm fraction and the agar fraction using LC mass spectrometry to measure the abundance of different phenazines that are made by Pseudomonas. These are the 3 primary phenazines that are made by the organism within these colonies. So here you can just see the distribution in blue in the agar and in yellow in the biofilm. Again, what I want to point out is the phenazines are chemically different. So they have different redox potentials, and they also have different charges at the pH relevant to the biofilms, which is circumneutral. So PCA is negatively charged, whereas phenazine carboxamide and piocyanin are neutral. Now, this impacts their retention in a very dramatic way. So if you look at the ratios of the concentration in the biofilm to the agar for these 3 different phenazines, what you see is that piocyanin is retained by far the best, and PCA really isn't retained at all. It partitions equally. And this can be reconciled through measurements of the binding constants for these phenazines to double-stranded DNA. This is from data done using isothermal calorimetry that I'm not going to show you, but here I'm just writing the binding constants. And there's this correlation where the tighter they bind, the more they're retained within the matrix of the biofilm that we know is chock full of eDNA. And you can do a complementary experiment where you take a mutant that can't make any phenazine and you can impregnate the agar with these different phenazines. And you can ask how much is taken up over a certain period of time. And you can see that the piocyanin phenazine is able to be taken up into the biofilm by far the best of these 3 phenazines. So we did a bunch of other types of experiments using genetic analysis where we manipulated the matrix of these biofilms to remove different types of polymers that are present, such as exopolysaccharides. And what we found was that those polymers didn't matter. You could get rid of them. And in fact, the phenazines were retained even better when you got rid of the polysaccharides. And so through both in vitro and in vivo genetic work, we are quite confident that it's the extracellular DNA that's the polymer in the biofilm matrix that is capturing these molecules. So this allowed us to then leap to a question that was a little more exotic, which was to ask, is it possible that when the phenazines are intercalating into the DNA, they're actually able to transfer electrons through this DNA, and that might this possibly, help facilitate charge transfer within these films? So the reason we were emboldened to ask this question is that we had a lot of great help to answer it. And this was due to the fact that Scott's co-advisor, Jackie Barden, my colleague at Caltech, is a world leader in the study of DNA charge transfer. And thanks to leveraging platforms developed after many decades in her laboratory, she's a chemist, and the work of her former postdoc, Edmund Say, Scott was able to do a convincing set of in vitro and in vivo measurements showing us that indeed phenazines could serve as agents of electron transfer through DNA. So here's just some data to show you this in vitro where it's very clear. So this system is simple in principle. It is using an electrode to which double-stranded DNA is tethered through a linker in such a way that we attach the phenazine covalently to the end of the DNA strand at the end, where it is incapable of directly interacting with the electrode. So any reduction that we measure, any current that we measure, must go through the DNA to the phenazine if we see current flow. And as another very important control, what we can do is introduce a mismatch into the base pairs such that we're comparing the amount of current that's flowing through a well-matched strand, here in blue, to the amount of current flowing through a mismatched strand, which Jackie's group has demonstrated is attenuated when you introduce a mismatch. The electrons are unable to flow as well through the DNA because the pi stacking is interrupted. Okay, so if you see an attenuation and mismatch, it's a very diagnostic signature for DNA charge transfer. So here what you're looking at is a cyclic voltammogram where we're plotting the current as a function of a potential sweep. And so as we go from relatively higher potentials to lower potentials, a reductive sweep, what you see is a huge amount of current flowing from the electrode to the phenazine that's tethered at the end in the well-matched strand. Now with the mismatched control, you see a big abrogation in current flow. And with the control where we don't have any PCN in the system, there is no current at all. And this is in the presence of oxygen. So what's happening is that as those electrons are flowing, ultimately the oxygen is quickly reoxidizing the phenazine, enabling an enormous amount of charge to be transferred under these conditions. So that was really exciting. And we were able to do similar experiments using the DNA from the biofilm matrix, using different types of charge transfer agents. And I'm not going to show you this data, but suffice it to say that you can collect very convincing data that within the biofilm matrix itself, a similar phenomenon is able to occur. So this leads up to the grand finale and the hardest question, which is, can we get a quantitative handle on extracellular electron transfer within these complex biofilms? And can we, taking what we've learned about the capacity for DNA charge transfer through DNA, speak to whether or not this is a process that might be occurring within these biofilms? So here's my former student, Scott, setting up these biofilm electrochemical devices to allow him to do this. This is under a controlled atmosphere where there's no oxygen present. So in this case, we're going to use electrodes to re-oxidize the phenazine, allowing for cycling to occur, just because it's a much more well-controlled system. And this system was one that we developed in collaboration with another terrific electrochemist, Dr. Lenny Tender at the Naval Research Lab in Washington, DC. And Lenny and his team are experts in studying electroactive biofilms from the marine environment. And so with his help, we were able to apply this to the study of Pseudomonas. So what you have here is, again, this chamber that has nutrients. We're able to control a suspended electrode. Here you can see it, that has what we call an interdigitated electrode array, an IDA at the tip. So I'm going to zoom into this increasingly. Here, what you notice is there's one comb that we call the generator, another a collector, and these we can independently control via potentiostats. And that allows us to play a variety of electrochemical games to make different types of measurements. So these combs are interdigitated, which means that we are able to have a big array of these electrodes upon which a biofilm can grow. And here's what it looks like. All right, so now you see on the left the generator comb, on the right the collector. And hopefully what you can see is that sticking out from each of these are different prongs. And this is microfabricated in such a way that they interweave. And what you have are 130 distinct small electrodes in the middle, forming 65 pairs of a generator and collector. And this is really useful because it amplifies the signal that we can get, allowing us to get a better signal to noise ratio. And we need that because these are really heterogeneous films, which you can see here by just the beautiful staining of the eDNA in blue and the biomass in red. So how do we parse these? Okay, here, if I'm just imaging a slice 10 microns up from the surface of the IDA, it looks like what I showed you before, these biofilms that we can grow in a flow cell that have tons of eDNA, lots of cells. And here you see the merge, same spatial scales as what we've studied before. Only now they're sitting on top of these electrodes. And so this is the surface of the IDA. And I'm going to zoom in here to one generator and one collector comb. And what you can see in red are the individual cells, and they're solvated by this beautiful cloud of eDNA. And so again, using different electrochemical approaches, I'm not going to take the time to go through all of the gory details, but what I'm going to do is show you one illustrative example. We can turn these combs off and on to ask different questions about the mechanisms underpinning electron flow within these systems. And so one I think accessible experiment I can show you is when we turn on just one of these combs, the collector, and ask, can we convince ourselves in these systems that the current, the metabolic heart rate, if you will, of the biofilm metabolism in these films is due specifically to the ability for these phenazines within these films to mediate charge transfer from the cells that are growing on organic carbon, oxidizing it. And then the phenazines are able to conduct and transfer these electrons to the electrode over these distances. And so the answer to this is yes. And we can see that when we plot the current over time for these biofilms. For a control that is unable to make phenazines, there's no current. If we add piocyanin back to this control, we see current, sort of slow heartbeat, if you will, of metabolism. And if we look at the wild type that can make its own phenazines, this is amplified and quite clear. And the reason that it's sort of this rise and fall is very nice. It's because, in fact, what we realize is correlated with is the temperature fluctuation in the room over the span of days in which we did these experiments. And that's reassuring because metabolism always responds to temperature. And so there were many other electrochemical experiments that we did that I'm going to not show you for the sake of time. But what I'm going to do is take the bottom line, which is what we learned about how the rate of electrocellular electron transfer in this film that we're going to call the apparent diffusion constant that we could measure through a variety of electrochemical tests using the system compares to the rate of physical diffusion or the loss that we saw of the phenazines from these films. And the bottom line is that within these biofilms, the electrons are able to pass much more quickly than they appear to be lost from the film. And so that's really striking and really important, which is that indeed they are preserved and recycled in a way that works, that's faster than how quickly they are lost. Mechanistically, now is the question, how does that work? And this is sort of the front edge of the research. And I can say that the jury's still out. What we know is that it's possible for this apparent diffusion rate that we're measuring within these films to be a function of both the physical diffusion of phenazines within the film, plus a term for what we call self-exchange. That is the ability of the phenazines to either transfer electrons to each other without diffusing, or transfer electrons to each other mediated through rapid DNA charge transfer. And it's not necessarily mutually exclusive. We could have some diffusion and some extracellular electron transfer mediated through DNA charge transfer. And so this is, again, the final summary. And I can tell you more about how we're hoping to distinguish between these. But what we do know for sure is that phenazines are retained within the biofilm by eDNA. This means that it's, in theory, it's possible for phenazines of different redox potentials to diffuse within these biofilms and just not be lost by virtue of the fact that they're almost capped by the phenazine with the greatest, the highest potential, piocyanin, being best retained within the system, serving as a net, if you will, a molecular net to capture these phenazines before they're able to diffuse away and help recycle them. And so, if you will, we envision a large cellular electron transport chain by these molecules within these films. And there's some direct imaging data that supports the idea that that's actually plausible, which is quite exciting. But what's also true is that within these films, it's possible that there is indeed direct charge transfer. It's just that we know these films are really heterogeneous. We don't have DNA, you know, in a highway connecting the cell in the internal part to the external periphery. And so it seems implausible that it's strictly DNA charge transfer. I really feel like it's most likely to be a mixture of the two, but that's a detail. And so what's not a detail that I think is important to keep in mind is the fact that these metabolites, these beautiful redox-active metabolites, are made by lots and lots of different organisms in many, many different flavors in many contexts, whether it's the soil in the roots of plants, the mucus of human infections, the sediments in the marine environment, or the sediments in an aquifer. Janet, this is the Haywee Reservoir, just for you. Um, and what I want to say is that I think this idea that these metabolites might sustain slow growth physiology in these habitats is one that I hope more people will start to, test to try to see if that's true. Because I think it's through the agathokakological reactions that these molecules are capable of achieving that they're able to sustain populations across a range of habitats, and that the adaptations that they've made to both the good and the bad parts of these molecules also help us reconcile how it is that when we find these organisms in contexts where we don't want them, like infections, we now have new insights and strategies that will help us better control them. So that's the bottom line. And thank you so much for the invitation. This is a photo of my lab from a day this past summer where we actually managed to get out there and do something fun despite COVID. And it's a great group. And as I've gone along, I've tried to acknowledge each and every person who's contributed, but we're working on a whole bunch of things. And if you're curious, I would direct you to the website where you can read all about it. So with that, I will end and be happy to take any questions you might have. And thank you all so much for your time and attention.